Today we're going to be looking at nerve and nerve impulses, and this is quite an important area, quite a common bigger question that you might get at A level for either AQA or OCA. So here we have first of all the structure of a neuron. So if we look at the right hand side, first of all, there are three basic neurons, as you should know from the reflex arcs lesson, and that is we start off with a sensory neuron, and effectively they've all got a cell body, they've all got a dendron or an axon, and that's really it. So the dendron is like the tube, or think of it as a tube at the moment, going towards the cell body, and the axon is the one going away from the cell body, and then obviously we finish at synapse at the end, and the bits where they receive impulses are dendrites at the end there. Then we've got the relay neuron in the middle, okay, or the intermediate neuron, and that is effectively a very small neuron cell body right in the middle with axons and dendrons either side, and then the motor neuron, the cell body is at the start, and the axon follows on. So you've got small dendrites in that case and longer axon. And what you can see here is you've got the different what a neuron looks like. So we've got this myelination, and we'll look at what the importance of that is at some point soon. It makes the nerve impulses faster though. So you've got this layer of fatty Schwann cells from around the outside of the myelin sheath, and if you were to cut that and look at it from a cross-section point of view, you've got the axon in the middle here, and then the myelin sheath or the Schwann cells are wrapped in like a sort of like a spiral sort of shape around them. Show you that there's some actual science behind this. You can see here an electron micrograph, or sorry, a light micrograph even. So you can see it under a light microscope, shows you the size of a motor neuron, and you can see there you've got the muscle fibers that it's attaching to. So effectively the impulse is going down this way to the effector, to the muscle fiber, and you can see just a different version here of a myelinated neuron to sort of show you that. Now, the important thing we're going to be looking at when we're talking about nerve impulses is that they are electrical impulses. So from now on, we're calling them action potentials, and they are electrical impulses that rapidly transfer along the plasma membrane of the nerve cell, along the axon. But when we're talking about electrical impulses, we're not talking about a flow of electrons. Instead, we're actually talking about changes in charge due to sodium and potassium ions moving in and out of the axon. So let's have a look at this and hopefully make a bit more sense of it for you. Here you can see a resting potential of the resting potential, how it's set up. So the resting potential is the word that we use when we say the nerve isn't firing an action potential, it's not firing an impulse. The cell is not actually at rest. Even at rest, the nerve cells need ATP, and we'll look at how in the next few moments effectively. But effectively what we call the resting potential, when the nerve is at rest, it's not firing a nerve impulse or an action potential. And the reason this is important is because we need to look at the charges. And what we say is inside the axon, so inside the actual nerve cell here, is slightly negative compared to the outside. Okay, so inside is negative compared to the outside, and you can see that you've got lots of negatives and lots of positives, but it's not that clear. We don't have a mixture of negative and positive ions. Both ions that we're going to be talking about are positive ions, so we've got sodium and we've got potassium. But effectively what we have is we have more more sodium on the inside. So let's say we had, and I'll give you some real numbers in a second, but let's say we had 100 sodiums on the outside, and on the inside our potassium is on, let's say, 40. You're going to be less positive on the inside, and therefore in comparison you're going to be negative. So we call this the resting potential, and we say there's a potential difference on the inside of minus 60 millivolts, and that is because there are less positive ions on the inside compared to the outside. And we say the axon at this point is polarized, and that will become important later on because we'll say that the axon becomes depolarized when we fire an action potential. Let's have a look at that. Let's try and make sense of this. Why is there this this difference in charge between the two, especially when we're talking about two positively charged ions? So here you have a different version. I much more prefer this version. So if you remember from the lesson we did on the preceding corpuscle, we said when an action potential in the sensory neuron is fired, what it does is it deforms these sodium channels here. So we call them stretch-mediated sodium channels. And effectively, what happens is these sodium channels are closed, okay, when the nerve is at rest. So the sodium gates are closed. That keeps the sodium on the outside of the cell. Okay, so this is the outside of the cell, and it keeps potassium on the inside of the cell. Now, to maintain this balance, because when the sodium channels open, all the sodium will rush into the cell, but they will only open when there's an action potential. There is something called, to maintain the balance, there's something called a sodium-potassium pump, which is a use of ATP. So remember those essays, what could be used of ATP? This is a big one. So what this does is it constantly maintains the movement of sodium and potassium, and it constantly moves in two potassiums in, okay, so 2K go in for every 3 sodiums that are leaving. Now that number is very, very important, because if you remember what we said a moment ago, we said that the inside of the cell is slightly negative, inside the axon is slightly negative compared to the outside. Well, look at that numbers, that sort of shows you why it's slightly negative. For every 3 sodiums you move out, you only push 2 potassiums back in. So let's have a look at the actual way this works then. So basically there are 2 types of proteins that act across the membrane of the axon. There are channel proteins that open, which allow the rapid diffusion of sodium or potassium through them, and at rest they are closed, and that is quite important. So that decreases the permeability. They do leak, so some will move across, but they are closed as a whole, so it stops the majority of the water going through. Think about a lock on a canal. It stops most of the water going through, but a little bit will constantly leak through. Then you've got sodium potassium ion pumps, which are the bits down here. And as I said, that uses ATP. So even at rest, the nerve cell is using energy. And effectively, that is actively transporting 3 sodiums out of the cell for every 2 potassiums that are going in. And that maintains then this negative charge. So this is why we say it has a charge of minus 60 millivolts. And effectively, that's what you need to know for the resting potential. How is that charge maintained? The sodium channels and the potassium channels, which we don't talk that much about at the moment, are closed, and there is a sodium-potassium pump using ATP, which maintains the negative charge on the inside, because for every 2 potassiums pumped into the cell, 3 sodiums are pumped out of the cell, and that is when we say the membrane is polarized. So here's just a different way of looking at this, and you can just see here you've got a potassium ion channels closed, so the potassium can't move out. Some does leak through, but we don't need to consider that at the moment. Different nerves will have different things to that. Sodium ions are closed here to channels, but this bit is where your movement is happening, so you've constantly got this 3 sodium ions moving out for every 2 potassium ions that are moving in. And effectively then that sets up this minus 60 millivolts resting potential. Okay, hope that's been useful for you guys. The best thing to do now is move straight onto the action potential lesson, which shows how this changes to fire a nerve impulse.